All right, welcome back, everyone, to part two of this special holiday episode where I'm here with Dr. Veera Rajagopal, who's a scientist at Regeneron, and we're breaking down some of the biggest stories of 2023 in the world of genetics and precision medicine. So we finished the last episode talking about founder populations and some of the major population genomics programs that are uncovering things that were impossible or going to be very difficult to uncover in some of the European ancestry-dominated research programs. We talked a little bit about FinGen, and we talked about a really interesting variant identified in a Puerto Rican population through some 23andMe work. We're going to continue the discussion around some of these population genomics programs around the world. And the first one we wanted to jump back into was a Notch3 mutation discovery in a South Asian cohort that you, Dr. Veera, highlighted in a, again, amazing tweet thread that we'll have in the show notes. Maybe you can talk a little bit about that one, why it was so interesting to you, and you flagged it up.
Yeah, so we are talking about findings from non-European population. So South Asian population is my favorite for the obvious reason. I'm from India, but also the South Asian population has like a lot of untapped potential for many genetic discoveries. Still we are just scratching the surface, and most of the GWAS studies, the South Asian populations are the most underrepresented, and there's a lot more to learn. What kind of genetic risk factors are there and things like that. And so this study is from my colleagues at the Regeneron Genetic Centers. So we collaborate with a lot of academic and research institutions across the world. So one of our collaborators is from Pakistan Genomic Resource, led by Danish Salim. And you might be familiar with this physician. He's a physician scientist, and there's like many landmark papers from that resource. One of the first human knockout papers came out from this resource. So here, my colleagues have been like looking into the genetics of stroke, and when they did an exome-wide association study, they identified one particular locus near Notch3. This is a very interesting locus because Notch3 mutations causes a Mendelian disease kind of, stroke syndrome. It's been known for a long time. It causes very early onset stroke, and also it characterized by, many other features like dementia and things like that. And I'm sure like when people started looking into, like stroke, when they first started doing GWAS of stroke, when they were looking into the findings, always when we do this kind of GWAS, we always go and look into our favorite genes or genes that we know like really, Mendelian kind of, that causes Mendelian kind of diseases, right? So, and interestingly, so far, there has been no GWAS signal at this locus. The last GWAS of stroke was like more than 1 million individuals, I think, like from predominantly based on European cohorts. And now we are looking at one particular variant, coding variant, that is probably the most important risk factor in South Asian population. And this is like really important because, for example, if we are doing a disease risk prediction, right, using genetic risk scores or anything, we'll be like completely missing out this variant if we are using an European-based GWAS to predict the risk factors. And also, this is like, and very clear, we know a lot about this genes being like, it causes this Mendelian disease, been investigated for a long time. And, and now we know there is like a mild penetrant mutation as well. And this is a major risk factor. Stroke is very prevalent in South Asia. So this kind of is reminiscent of what we found in African populations, African Americans, for the APOL1 kidney disease, right? So it's like a very clear target population, and you have a gene, and then, there is a huge opportunity to make some big, translational impact, in these populations. It's a very similar situation here. That's why I'm very excited about this finding. And I'm sure like there are so many findings like this. This is just, the beginning of all this, important discoveries that will be coming from the South Asian population.
Yeah, and there were a couple of really interesting things that stood out to me from the paper in this finding. I think the size of the population was around 75,000 Pakistanis who were sequenced. So first of all, orders of magnitude smaller than the million person GWAS that you've described before, but also great to see that some of these exome sequencing programs are getting to scale in places besides the UK. So I suspect in the next couple of years that kind of resource may reach hundreds of thousands of people, which is great. And also, I think they estimated that around 5.5% of strokes within this population could be attributed to, or at least have that particular variant in that gene as a major causal factor, which is pretty interesting in and of itself, that you're explaining a large fraction of a very common issue where there was really no clear genetic evidence before. So I think it's a pretty amazing piece of work that highlights both the underlying biology, but also seems like it could have a very clear and immediate impact within this population as well in the next couple of years. This is the kind of thing that you can think about screening for in families with a family history of stroke, for example, as part of a broader panel. So I think this is the kind of work that hits discovery, translation, and healthcare pretty quickly, right? You could see this having an impact on people immediately and not just being a research finding that's kind of curious.
Absolutely.
There's another paper that we wanted to highlight from South Asia. So there was, I think, the first probably large-scale whole genome sequencing data set that did an interesting look at the genetic architecture of South Asia more broadly. So 5,000 whole genomes published, I think, in June of this year, or May, actually. And you wrote a great thread about it in June. Maybe you can talk a little bit about that and why this paper was a landmark.
So the reason why I was telling before, the South Asian population, has like untapped potential for discoveries is like two major population phenomena, right? So one is like consanguinity and then endogamy. So these are cultural practices that's been happening for thousands of years. And endogamy is like, even a bigger one because of how the caste system has been organized for, since the colonization or even before that, but it intensified after the colonization. And these actually had a stronger impact on the genetic architecture of Indians like that's living today, right? So we talked about Finnish population, we talked about Icelandic population. These are all bottleneck population. And as a result of, like isolation, people have been marrying within this community, and this results in the increased frequency of rare deleterious variants, right? And so that also increases the opportunity to identify homozygous variants who might be a knockout for a particular gene. So now imagine like Finland or Iceland, but like 3,000 to 4,000 Finland, Iceland packed into a, country. Right. That is India. So right. This is like you have so many castes, so many groups, and, like there has been like some landmark studies on this that have clearly shown you can actually quantify how much of, parental relatedness is there based on the genome, based on looking at the stretches of homozygous segments, right? Regions of homozygosity. But previous studies has been very scattered, and they use this, like DNA array data to look at some of the common variants. And, and this is the first time we are actually looking at the rare variants and the exome sequencing data. So this is from, a company called MedGenome in India. It's like an emerging company. And also they have collaborated with companies in US and also a consortium, a big Genome 100K Asia consortium from Singapore. So they have accumulated samples like 5,000 whole genome sequences comprising of Indians and also, Bangladeshis and also Pakistanis. And there's like, again, they find all this, very high prevalence of loss of function variants, and then very high prevalence of people who have homozygous loss of function variants. And the very interesting thing is like when you actually look into the, like the origin of these mutations, right? So the enriched loss of function variants in India, for example, is unique compared to enriched loss of function variants in Bangladeshis or in Pakistanis. That makes sense because like these enrichment happen within small communities. So now if you can zoom into this, for example, India, and you look into individual populations, individual, caste communities, each caste community or the genetic clusters will have their own enriched loss of function variants. And often the genes with homozygous loss of function variants that you will see in this group probably is the only place in the world you will see. So we need a map of loss of function variants enrichments across the, the country. And this will not only help the people in the community, about what kind of diseases they get from these mutations, and helping them genetics testing, screening, and also but also it helps a lot with drug development and things like that. And that is why people are really interested into this. And this is kind of, we get all these insights from this very beautiful paper. They looked into this, and it's really nice. I recommend people reading it. But this is like, as we go, in the upcoming years, we'll be able to see more of this, all the human knockouts, loss of function variants, how much insights we get from these carriers.
You have a sense of how big a data set like this needs to be for you to start to discover 80-90% of genes knocked out in its entirety? Because you made the great point earlier. There's both the public health and medical benefit to this, but also we have a long-standing problem in the field of we don't know which genes, what happens when you homozygously knock out a gene. Is it compatible with life? Is it cause rare disease? Does it contribute to a common disease? What is essentially the impact of this? And I've seen simulations that look at populations in Europe and then populations that may have higher rates of consanguinity where you'd see this at a higher rate. Do you have a sense from the paper or from other reading that you do of how large a data set like this needs to be to start to, maybe saturate isn't quite the right word, but to start to get a coverage of every gene in the genome and have some sense of what happens basically if it's knocked out on both copies?
Yeah, so that's a very good question. So there's like a lot of challenges about these kind of estimations because of the huge heterogeneity, right? So, and I really think the initial step would be to have, a survey of what kind of mutations are occurring more commonly in which regions of, the South Asia, right? And then you can then, if, for example, there's like a big discoveries coming up from European-based studies or some important drug targets being developed, and then we can come back here and then, you can see, hey, this gene mutation is enriched in this region. We can do like, targeted sampling of that region to do a lot of studies. So that is how it should be, right? So then you can, based on that, you will know how many samples you need from this particular region. And this, for the same reason, is also difficult to have like a single imputation panel, like we have for other population that is going to work across the country. And that is a other big challenge, So because like I was mentioning to you, Finnish population, FinGen biobank, Icelandic biobank, they did not opt for sequencing, right? Because they know they can find 90% of all the enriched variants just by sampling a small subset of the population, doing a whole genome sequencing and imputing the variants in the rest of the population, which is not going to be the case in regions like India.
Yeah, I also noticed from this paper that they developed a array, I think they called it Sargum, that was specific to this population as well, which was cool to see because we know that there are biases in most of the commonly used genotype arrays, right? To sort of have one that doesn't miss some of the more common and high impact variants that could be totally absent or hard to impute in some of the other arrays was also good to see.
Yeah, that's a very good point. I think that is also available from the Michigan imputation server. If you were, doing any South Asian based GWAS and you have genotypes, then you can actually impute using that. And on that point, now we have even a larger imputation panel from UK Biobank. The UK Biobank have 10,000 whole genome sequences of South Asian. That is the largest to date. It's very small compared to Europeans, but it's the largest to date. But it will be really interesting to compare how this panel compares to a panel that comes from India, right? So I'm sure there'll be like a unique variants that can be, so that like probably someone would want to combine these two panels to make a other updated panel that will really help, I think.
Yeah, because the point you made at the beginning of this around endogamy as a cultural practice, this is likely going to change as people migrate around the world and live in different places. The effect on the genome will take generations, though, to change, won't it? But it will be interesting to see to compare and contrast large studies in India, Pakistan, Bangladesh with the same people from those countries that have emigrated to other places in the world to see what differences, if any, and may help us also to tease out differences in environment and gene interactions, right? As we think about with the same genetic ancestry, but living in different parts of the world and different environments.
Yeah, definitely.
Yeah, and maybe that's a good segue actually to the final paper in this section, which was a paper by Hillary Martin and Daniel Malosky, who I know both know very well. We worked together in Matt Hurl's group for a while. Hillary and Daniel and I went actually university together, but they did this great paper looking at the impact of consanguinity, so marrying close relatives, and the impact on common disease risk. We spoke about this before, but you have a increased risk of some recessive rare diseases. That's probably clear why, because you're more likely to inherit two copies of the deleterious variant. But there's been an open question for a little while around what impacts this might have on common diseases where the risk is more complex or polygenic. And I don't think they touched too closely on this gene by environment interaction question that we were having here, but there's a natural extension here to think about layering that on top. So I wonder if you could talk a little bit about that paper and why you picked it.
Yeah, this is another fantastic paper. It uses the samples, South Asian samples, British Pakistanis from the genes and head, biobank that is in the UK, and also from the UK biobank. So the question here is like, we know very well that consanguinity that results in increased, mutation load of recessive mutations in the offspring actually have an effect on, it causes a lot of rare diseases, skeletal deformities, like intellectual disabilities, neurodevelopmental disorders. And this has been known for a long time. And the question they ask here is like, these increase in mutation load, recessive mutation load, has an effect on complex, common diseases, common and complex diseases. This is really a very interesting question, because if we go back to the GWAS literature, right? So most of our GWAS association studies is based on linear additive model, and so the most contribution comes from additive effect from the variants, right? And that's been like always a big interest in knowing what is all that dominant risk alleles or all that like recessive risk alleles. I think for type 2 diabetes, there is like really cool 2 or 3 papers that have been like from the Broad Institute, where they have did recessive association tests and identified some mutations. But the thing is, like, generally there is interest. This kind of here, what they are showing really answers partly that you need population like South Asian population to answer that question. And people have been looking into the wrong population with that question, because there's like in the outbred population. So what the authors here found is like, they found that the increase in autozygosity, the increase in mutation load, is associated with increased risk for common diseases. There is a very big problem here when doing this kind of studies, is because these are cultural practices that is what have shaped the genetic architecture, right? Consanguinity and this marrying within the community. And so this always go hand in hand with all other confounding risk factors like religiosity, or having a very particular kind of diet, or addiction to very specific substances. So it's very, it's challenging to disentangle that, because otherwise it's down to the, our basic issue of population stratification, chopstick genes, identifying chopstick genes, right? But they beautifully solve this issue by looking at very, highly consanguineous, people who are offspring of parents who are highly related. So when by looking at these only, people with very high consanguinity families, they kind of avoided this confounding factors, but they also actually did some within family studies. Even within family, the siblings, the amount of deleterious mutations they receive from their parents can mildly differ, and you can leverage this difference to make these kind of association analysis. They actually did in the 23andMe, and they showed that replicated the finding. So the most they identify like few diseases, they highlight type 2 diabetes because it is very highly prevalent in India. And we talked about a paper last year, right? So, and they show here that just the recessive mutations, right? This kind of recessive mutation load alone could be explaining more than 10% of the type 2 diabetes cases in the Indian population. And that is the kind of question people have been trying to answer by looking at the European population, looking at dominant and recessive, alleles. And so for many reasons, this is really a fantastic paper, actually.
Yeah, and where do you think they take this from here, are there big unanswered questions that will be addressed by having some of these large scale whole genome sequencing data sets outside of European ancestry populations, or do you feel like they've covered quite a bit in this paper and there's something else, something different?
I think like the next interesting thing would be to go zoom in, right? I mean, this is like genome-wide statistic. So really zoom in to identify what kind of pathways, disease pathways or genes are acting through this. We know only a handful of recessive genes that was for type 2 diabetes risk. So that is the next step, I would say. And for that, you will need either large sample size or, like we wait and then there will be like isolated reports, case reports from some clinic or someone. This is like like some very highly penetrant mutation causing MODY or some other gene. And then you connect it back to this paper, right? So somehow the puzzle will start fitting together as years go by. But that is, I think, the most interesting thing, because it'll be very nice to know how these pathways differ from the ones that we identified based on GWAS studies, and if they will help with any of the drug development efforts.
Yeah, absolutely. I'm interested to see as well whether there are ways they can tease out this gene environment interaction question a little bit more. It's a very tricky one, but you're right. Actually, they managed to do some really elegant work by looking at the within sibling analysis from the 23andMe dataset. Okay, so moving on to our next topic here, we want to talk a little bit about some of the maybe less low-hanging fruits. There were many genes that were discovered early on in the GWAS era. We talked about APOL1, FTO, BCL11A that could be discovered with a relatively small number of people. And those have been some of the stories over the last couple of decades of working out the biology underneath those. But we've actually had some things that maybe could have been described as low-hanging fruit, but for some reason weren't discovered until relatively recently. And maybe you can talk about one of these examples, which is a gene called, or at least a locus near a gene called ADR-A2A, which plays a role in Raynaud's pathophysiology. And maybe this is actually a good one to talk about the breaking news that we were talking about before, because that might actually fall in this category as well. But I'll let you talk about this paper first, and then we can decide if we bring that one forward.
Yeah, sure. So this is one of my favorite theme where, so the GWAS era started like in 2007 or 8, right? So after the Human Genome Project, we had this, this linkage disequilibrium based imputation has picked up, and then we had this people started doing GWAS, right? So they picked the traits. They picked the most common traits: type 2 diabetes, type 1 diabetes, hypertension, bipolar disorder. And interestingly, many of these traits, immediately showed up genes. These genes they explain a lot amount of heritability, and you only need like few hundred samples or sometimes even less than 100 samples, for example, for the AMD, CFH locus. So the important question here is like, how many such locus are there? I mean, these are the diseases that we know we've been like, we are really interested. So they picked it. And there is like thousands and thousands of diseases if you take the ICD chapter code, right? And there should be genes that are like really important, like that you can identify by doing just a GWAS of, let's say, like 1,000 samples or 500 cases or so, right? And that kind of studies often come every year. I closely watch them. We had I think a couple last year. And this year we have this one particular paper, and it's a very interesting condition. They did a GWAS on a very interesting condition called Raynaud's phenomenon. It's been like, it's characterized by, vasoconstriction, sympathetic activation when the individuals are exposed to cold or stress. It's not very, pathologic. It's self-resolving, but it can be very discomforting. And very interestingly, this has been very first time described by, so it's named after the person who described it. It was a PhD thesis project. So I don't think it's even published. It was like based on a thesis project that he did, and Maurice Raynaud, I think the name. And okay, but what did they find? They find this particular locus, very strong locus near the receptor, adrenergic receptor, right? And the beauty here is we very well know this kind of very well aligned with the pathophysiology. We know it's the sympathetic nervous system is what is like being affected. And here we are finding, the adrenergic receptor. And this is known for a long time. I mean, like even for like in 90s, they have shown using experiments by giving adrenergic blockers, specific receptor blockers, and see which blocker actually, release the Raynaud phenomenon. And it's like it's this what they find here is exactly the one that they demonstrated previously. And the here, but it also adds in another like the subtype. So previously it was like mainly focused on ADR82C, but it seems based on the GWAS, the ADR82A is probably having the much important role in Raynaud's phenomenon. So this is like a very satisfying finding, but also it's something that you could have found if you picked this trait like in 2007 or 8. It's just you need like so very few samples. This is like reported by two teams. The the striking thing is like one team, the team from the Finland, they actually did this analysis. They used the Finnish samples, they used the UK biobank, then the couple more samples. And there is like you will see the picture, the plot in the paper. There's like 4 Manhattan plots individually in each of the cohorts have genome-wide significant hit for ADR82A. I don't think I've seen ever like that ever before. You
Yes.
like do the meta-analysis and then you get the significance, or you have a genome-wide significance in one paper, then you replicate it like 0.05 or something. It's like
Right.
4 independent samples. Everything is giving genome-wide significance. It's such a strong locus, and it's like amazing to see. We didn't know this all these years.
Yeah, so I guess the interesting point here is why did something this strong and go undiscovered for so long, and also how many other are these lurking out there? I was thinking as you were saying this, so they did this in 4 different biobanks, as I think you mentioned: UK biobank, Estonian biobank, FinGen, and then I think there was one more. Were there anything that changed in these in the last 2, 3 years that meant it couldn't have been discovered before? These are common variants. It's a phenotype they'd known. Is it a question of just there are so many phenotypes to look into and not enough scientists? What's the rate limiter for us finding? Because you're right. I mean, you could think of anything and everything that you could ask on a questionnaire or that you could pick up in a medical record. There may be low-hanging fruit to discover. And this is the first time I've thought about the fact that maybe there are some really obvious ones that are, maybe it's not obvious, and I don't want to disparage the great work they've done, but this is a kind of GWAS that maybe could have been done 10 years ago that nobody did, and I wonder why.
Yeah, so I'm sure there are like so many different, like disease traits like this, and it's just a matter of, like which one you give importance to, right? So, and also someone to take notice of this, right? So I'm sure like a lot of time when you do like people nowadays do like phenome-wide association, right? Every time your biobank comes up, all of us or UK biobank, let's do an association analysis across all the phenotypes that we have, all the ICD codes. And sometimes you also find this in that kind of scan. But I think that it also it's a lot of time you will find only when you actually you are looking for it and this particular trait. And for some reason, this trait might not be even included in your phenome-wide association analysis. So it's a very interesting kind of theme that I really like to, like I watch for like this kind of findings.
Yeah, and maybe this would be a good time to talk about this interesting late breaking story. So we're recording part 2. We took a little break in between part 1 and part 2, and this story came out in the meantime. But I know it was on your radar already. This seems like it's maybe in the same category actually of the gene had been studied for some other phenotype, but then very smart scientists put 2 and 2 together and linked it to something that was previously maybe buried in the literature. I wonder if you could talk a little bit about this, and we can cover it now.
Yeah, so just a few days ago, there was a paper in Nature from, scientists from MRC. Some of them are like endocrinologists, very big shots. The last author, Stephen O'Reilly, he's a famous obesity research. He's the one who actually mapped some of the very first monogenic Mendelian conditions of obesity. And so really endocrinology, genetics is, I really love endocrinology. It's very fascinating. Often the mechanisms will be like really, really fascinating to look into. So what they found here is like there's like this GDF15. It's a cytokine, it's a hormone that is actually increased during pregnancy. And they found this is actually the major protein that is responsible for a condition called hyperemesis gravidarum, which is characterized by very severe nausea and vomiting. Like a big proportion of pregnant women actually suffer from this during early trimesters, like early part of the pregnancies, and this sometimes can be very severe. The first author, actually, Marlena, in this paper, she actually also had this condition, and that is one of the motivation for her to like to study on this particular condition for her, like, in her whole career. It's a very interesting story. So it's a long evolving story. The GDF15 protein itself was found as like in 1997, I think. It's some component of serendipity in that, like they were looking into proteins that's been associated with macrophage activation, and then they found some very new protein that they never saw before, and they got interested into it, and then they cloned the gene, and then only they found the GDF15 gene. And so so these people, I think it's from the people, Australian scientists, they've been like following up from then on. So they found that then they started looking into which tissues are being expressed, and they found that there is like this RNA, this gene is like abundantly expressed in the trophoblast, in the placental tissue. And that kind of led to them to study, is this like gene is highly expressed, is this increased in pregnancy, like condition. And they found, yes, this protein is like really high during the pregnant condition. And this is like, it probably secreted from the fetus, and this could be an evolutionarily evolved response for the fetus, because it's a cytokine to fight against the pro-inflammatory cytokines that's been produced by the mother. And so that is the starting. But then the story really evolved over the past 25 years, I think. So the one of the main early finding, it is again from the same scientists who cloned the gene. They found that this, when they were looking into what all the tissues has been secreting, they also found it's been like highly secreted by cancer tissues. And so they looked into the cancer patient, and the cancer patient GDF15 levels is high. Why it is high? And then they've been like looking into that. They found that GDF15 levels is actually is what causing this patient extremely aversive to food intake. And this is kind of causing them, for some reason, making them to, like lose their appetite. And this is one of the reasons contributing the cachexia, like condition that you see, right? And that actually resulted in very big interest. This is like a protein which actually make you eat less in loss of appetite. So it's obesity target. So that kind of, so I think many company, particularly industrial researchers, took up this, and it's been like intense research. And in 2017, that is the biggest breakthrough in this whole story, is like they discovered the receptor for GDF. And I still can't believe that 4 independent research teams from 4 different companies actually found it. They independently discovered it, and they all got published side by side in Nature Medicine. The most important revelation of this story is like the location of this TDA15 receptor. Okay, so it's located in regions of the brain stem, which are, involved in this something called emergency pathway, where, like it has like centers where if it is activated, this kind of prevent the humans to, like eat, like this is kind of probably an evolutionary response to prevent the humans to eat like toxins or things like that, right? And it's also the region that also has this kind of the chemoreceptor trigger zone, the emetics, the drugs that we have typically act by hitting this region. And also it seems that the increased vomiting and things that are also associated with chemotherapy also related to this region. Right. What happened is like Stephen was one of the person who actually got this, where in the presentation when it happened, even before the publication, and they were telling about the location, and he is like been interested in all this, right? And he remembered about the GDN of the 15 association with the pregnancy, and immediately he got interested, and he kind of predicted that this should be something to do with increased vomiting in, like in pregnancy. And then he started working on it. And this is what was like, so he started putting the puzzle one by one. And this is on the one side. On the other side, like I said, the first author of this paper, she has been suffering from this condition. And so she's a scientist, she's a geneticist. So she thought, okay, no one is studying about it, right? So that's other important fact. So like I said, the initial discovery is like they identified that it's increased in pregnancy, but for some reason, for the next 20 years, no one like really keenly looking into why it has increased in pregnancy, but people are like crazy about using this to identify targets to treat metabolic conditions, obesity, etc. So Marlena, she wanted to study this, and she did this GWAS of hyperemesis gravidarum in 23andMe with collaboration 23andMe, and she found that strongest locus was GDF15, and it was like really very interesting. And she also found that there's a signal near GFRAL, that's like the receptor gene, right, for the GDF15 when looking into the severity of hyperemesis gravidarum. And it's very well fit with what's been known, and like that's how they kind of like Marlena and Stefan, they kind of met each other, and those teams then they collaborated with a lot of other people. There's like many scientists in this in the story, and they kind of put together. Okay, so the major revelation, what is the major thing about this paper is that all this means like all these puzzles is getting put together, and now at some point, okay, we know that GDF15 is increased during pregnancy, right? And there's a spike, and this because like it's been secreted from the fetus, and that is like hypothesized before, but they actually for the first time, I think, like they empirically show that, and that is fine, and it is a risk factor, and it is causing the hyperemesis gravidarum, and there is a genome-wide locus for GDF15. But there's like one part of the puzzle that doesn't fit well. That is, when you look at the genetic variations associated with this hyperemesis gravidarum, it said that people who have genetic predisposition, right, genetic variants that increase the GDF15 level, right, they are actually at lower risk of hyperemesis gravidarum. And so Marlena also worked on an exome-wide association study. She actually worked in collaboration with Regeneron Genetic Center, who actually helped us sequence the samples she collected. And in this, there's like not a large sample, but they identified one deleterious missense variant, which they predicted to be deleterious. At that point, they didn't know, but it was like it increased the risk for hyperemesis gravidarum a lot. And later they found this is actually, like it prevents the secretion of GDF15. So it's associated with very severe deficiency. And what it says is like, when you have a rare variant that really causes very low level of GDF15 in you, it actually increases your risk for hyperemesis gravidarum substantially. So common variant finding, rare variant findings, both did not fit well with our known biology, because more GDF15 actually causes, higher risk for hyperemesis gravidarum. But Stephen, being an endocrinologist, his intuition was that, okay, if this is the case, then there is a possibility that GDF15 system could have like a ligand-induced desensitization, might be there. That could be the reason. Like this is the one that we also see in a lot of other hormonal systems. And so he tried to, see if that is the case. And it turned out that's actually the case. So they did mice studies. They primed the mice using low dose GDF15, and then they gave an acute bolus injection of GDF15. It actually did not cause any, did not reduce the food intake. That is a very established effect of GDF15. But if you don't prime the mice with low dose injection, it actually had an effect. It loss of appetite, reduced food intake. And that is like animal validation. Then they also did a human validation. To do a human validation, basically what you will need is like a naturally occurring genetic variation that raises your basal GDF15 and non-pregnant state GDF15 to very high levels, right? So those people probably are immune to hyperemesis gravidarum. Unfortunately, at least so far, we don't know that there is no such variance, right? So, but it turned out there is a rare Mendelian condition, thalassemia, right? So people with thalassemia actually have very high levels of GDF15. Like I mentioned before, GDF15 is produced by many tissues and cells, and it seems the red blood cells also, like hematopoietic cells also produce this under stress conditions. And so they use this as a system to see, then if it is true, then women with thalassemia should be immune, or at least have a very reduced risk of hyperemesis gravidarum. And we know that the fertility is very low, right, in people with like probably homozygous, but at least in the, like a moderate penetrant conditions, people do get pregnant and they give birth, right? So they looked into women with thalassemia, and they found that impressively less than only 5%, less than 5% of the pregnancy actually had any kind of nausea and vomiting, which is compared to more than 60% in the general population. So it's amazing, but most important thing is this might end up in a very, treatment for hyperemesis gravidarum. They have clearly shown GDF15 is a target, and also that its receptor, GFRAL, is a target. Like there's multiple ways you can treat. So one, you can actually do some kind of vaccination like approach where you actually, sensitize desensitize the women before they get pregnant. If like they might get it, like their family history of, like severe nausea or vomiting, something like that. But you can also use something like antibodies to GDF15, or you can even prevent GDF15 to go and act on the GFAL by, knocking down or prevent, like blocking the GFAL. There's like so many different pathways, therapeutic pathways, and this like I'll closely watch this. It's going to be like really amazing story how it ends up. Like I think it's a fantastic story. And the other thing is, like you said, you put this correctly in this topic. Like it's the number of cases Marlena used in the 23andMe collaboration. Like very few, like 2,300 cases or so. So again, this is a phenotype that could have been GWAS long time ago. And if it happened, the whole story this I told could be very different, because imagine GDF15 and G for 2007 or 2008, like that could have actually led to the discovery of the receptor for GDF15. Right now, the GWAS locus said, oh, this is the receptor for GDF15. If you found this like 10 years or 15 years ago, the story could have evolved differently. It's really fascinating to think about it.
Yeah, it's amazing stuff. And I think, at least according to the NHS website here in the UK, 8 out of 10 pregnant women feel sick. There's a spectrum here, some extreme nausea and vomiting, some a little bit more mild, but the potential impact of some of the different therapeutic approaches you mentioned here is enormous. And yeah, I was just thinking about this, low-hanging fruit question. It seems like there's a couple of important ingredients. You obviously have to have the data set to begin with, and we've talked a lot about different data sets that are being generated. The UK Biobank, 23andMe, and many others have existed for a while, but now we're starting to get much better representation. The second thing you have to have is a motivated scientist or group of scientists to go after that phenotype. But then I think the third one that this story highlights as well is a person or group of people that really can link the deep clinical knowledge to the understanding of genetics, because it's not sufficient to just find the hit in 23andMe or find it in the UK Biobank. This is some really, and I think one of the stories earlier, BCL11A is a good example. And one of the other ones that we discussed earlier, APOL1 maybe is a good example of this. There's an FTO actually was the one I was thinking of, someone who really digs into the details to try to understand some of the apparent paradoxes that the GWAS hits often present. I think that final piece is not to be overlooked as well, because you can't just solve this with having big data sets. You need people to go in and scratch their heads against some of these paradoxes that initially show up and try to figure out what's actually going on. So I think this story ties all that together really.
That's a fantastic point. I mean, like even if retrospectively we do an analysis of what kind of GWAS targets actually succeeded in terms of our understanding, then we would find that the ones that people, not just geneticists, but people who worked with like translational, with clinicians and like molecular biologists, who all work together to solve the problem. And yeah, I think that's the one important take home from these kind of studies that you should.
Yeah, and a couple episodes ago, episode 112 with David Ochoa from Open Targets, he mentioned that they looked at how genetics impacted the likelihood of a drug succeeding in clinical trials and ultimately getting approved. And they look retrospectively over many years. And they found something that I thought was really interesting, which was if you do a first pass and you don't look into the link between the genetics and the target and the drug super closely, you don't find a very high hit rate. But if you have somebody who really understands the disease and the biology, and you take that time to actually go in and understand the link between what genetic data exists or existed at the time and the finding, that you actually find a very high number of these. If you have the right person looking at it, they find really clear supporting genetic evidence. And it seems like it sort of fits with this model as well, that we have no replacement right now for the really strong clinical background. It has me wondering at what point will large language models and some of these improvements allow people who don't have that deep clinical understanding to start to harness that knowledge themselves. At what point do we have a Stephen O'Reilly in a box, a chat GPT? That I suspect we're probably far away from that, but it's one of the things that these models are good at is ingesting enormous amounts of knowledge. They're also very good at hallucinating and making things up, but it has me wondering at what point this becomes something that is available to everyone as well, but maybe not soon. Should we transition on to some interesting rare variant discoveries? And actually,
I think we're still in the same theme of kind of low-hanging fruits, but being like looking at the common variants, very similar theme in the rare variant.
Yeah, and a similar group of people are involved with this paper, at least Stephen, who you mentioned before, but a wider author list on this. Maybe you can talk about this new gene that they discovered called bassoon, which is a very nice name for gene.
Yeah. So last year when we have been like discussing about many rare variant discoveries, right? GPR75 and things like that. And that is because that's when the UK biobank exome sequencing just got out, and like people are doing all these discoveries. And so only this year, actually, I think people started like getting access to the full exome-wide data set, full UK biobank exome sequencing data set. And I've been like, I was like expecting to see many more, like associations, but I'm not sure if I actually did. But this one particular association is very interesting because, many teams found it. And firstly, like this is for the BMI, right? BMI, I think, is one of the most studied traits in the UK biobank. I think like there should be like at least 10 papers on people looking at the rare variant associations. So this kind of slipped away in the radar of, like RGC, or maybe but they later maybe did, I'm not sure, but it was not reported in the paper that they, published in the Science. And because they looked into like kind of burden associations less than 1%, and then people have been like doing analysis in different ways, looking at very, like severely damaging rare variants, very rare variants, right? And so this came up, and like not one from, like two or at least three teams. And they found when you look at this very rare loss of function variance burden, there is like this gene BSN3, where people, or like the effect size is really huge. It's even more than the effect size. You look at like heterozygous carriers of mutation, like in the MC4R gene, which is a very well-known, established, Mendelian obesity gene. And this is really impressive because, it's interesting that we didn't know about this gene all these years. Like MC4R been like discovered maybe like 20 years ago or so. And you can call that like a low-hanging fruit in the rare variant space, right? And we are finding some kind of intermediate hanging fruit, I can say, like. And it's very interesting because doesn't look like, what we typically see in the Mendelian obesity genes, like this open leptin melanocortin, pathways. This is kind of looks like a neurodegeneration gene. It's very interesting because this gene has been also implicated. This actually, how they initially discovered this gene was by looking at the transcripts, in the cerebellum of Parkinson's patients. So at that point, they didn't know anything about this. That's how they first found this gene BSN3. So now they find this association with obesity. And interestingly, just a few weeks ago, there was like a paper on early onset Parkinson from Indian population. Actually, that would have been another paper in the list if I looked into all my tweets properly. So now I remember it. So one of the genes that they found with the Parkinson is like BSN3. So it's very interesting. It brings neurodegeneration and severe obesity in together, right? So I'm really looking forward to see how this gene pans out. I mean, this is a preprint from MRC, but there's also a publication from Wendy Chung and other people from Columbia University. And interestingly, like one of the like disadvantage, or like from the UK biobank or things like that, when you actually look into the association, you cannot go like look into, for example, if you ask a question, how's the appetite of these people, all these people, like, and those questions you cannot answer because you are restricted the phenotype, right? Actually, this team from the Columbia University, they scanned the exome genome of a cohort of extreme obesity. They identified two women and with the loss of function variant in BSN3. And it's like they have extreme obesity. One is like one of the patient was an African American woman who had like 60 plus BMI at the age of 15. And the other one also like had extreme obesity because of severe hyperphagia. And both of them had gastric bypass surgery. It's the kind these rare patients actually give little more details on patients carrying this mutation. And this is really interesting. And I don't think those patients had any kind of evidence of neurodegeneration or anything. So there is something happening with the how the mutations results, right? Whether it's results in obesity or Parkinson's disease, or it's going to be very interesting to see how this pans out in the upcoming years.
Yes, and I was going to look it up right now because I remember reading this Parkinson's in India paper, and it actually caught my eye because of the novel variants found in the GBA gene, but I hadn't realized that they identified variants in bassoon as well. Do you remember if they were loss of function variants in bassoon or not? Because I think one of the neurodevelopmental phenotype is associated with missense variants. I think the obesity with loss of function, and I wasn't sure about the Parkinson's disease, but clearly there's maybe gain of function and loss of function kinds of things going on.
Yeah, I think it's a loss of function, PLOFs, or maybe PLOFs and deleterious missense combined. It's kind of like,
Yeah.
it's burden test. But yeah, it'll be interesting to see. There is actually a mice knockout. And when you actually create a homozygous, like, full knockout, actually the mice have like neurodegeneration, spontaneous seizures, but not obesity. So it's here, what we are looking at here is heterozygote carriers who have severe obesity. And probably the homozygote full absence of this gene is lethal, I think. So probably it's very rare to see, but probably we might see at some, we might see some report of, consanguineous family having one member or someone, with this knockout, and probably that will give more insight. So yeah, something to really closely watch for.
Yeah, why do you think this gene was discovered so long after MC4R and some of the others? You mentioned them having comparable effect sizes. I think just for people's reference, the effect size of MC4R is something in the range of, I think, 2 kilograms of additional body weight per carrier. This one is between 2 and 2.5, I think. So it's a big effect in and of itself. I'm wondering why it's taken us till 2023 to find it.
Yeah, so I think it's the rarity of the variant. The MC4R is relatively more common. We have more number of carriers than, right? And it's also like MC4R, I think, is like you might be enriched in a lot of populations. And you also have, it falls in the very well understood biological pathways. So, that kind of makes you go look for it, right? And yeah, regarding the effect size comparison, definitely it's bigger than MC4R in the analysis, what they did in the UK Biobank. But also I need to highlight the fact that people with MC4R mutations in the UK Biobank is kind of slightly biased towards people having a less severe form of the mutation, because of some modified penetrance. And there are people who also have like, more severe forms of obesity if you look outside the healthy population. So yeah, but still it's really amazing to see this bigger effect size than MC4R in the UK Biobank.
Yes, no, that makes sense. I thought maybe it'd be worth us talking about a couple of these non-coding variants, actually, because this is an area that's close to my heart, having spent 4 or 5 years of my PhD looking very hard for non-coding variants that cause developmental disorders and not finding very many. But they are out there. And I remember reading papers of some amazing work looking at enhancers that cause all sorts of rare problems in rare disease cases. But there are 2 that came out this year, one in FOXA2 deletions of in FOXA2 and another zinc finger. Maybe you can talk about the first one and why it caught your eye.
Yeah, so this topic is probably also very timely, because with the release of whole genome sequencing, there's been so much interest in what people are going to find with the whole genome sequencing, right? That people didn't find with the whole exome sequencing. And one of the first thing people often say is like non-coding variants, like non-coding rare variants. Also, I like this topic very much. I highlighted few examples of very fascinating non-coding rare variants, but they saw those causes Mendelian diseases. Again, it's very similar here, right? So I think the general thing we all agree is like we are just scratching the surface of how this non-coding variants causes disease, but like it also limits our understanding of their structure, right? They don't have like we don't know like we cannot annotate the variants like we annotate for the coding variants like loss of function or missense. And so for many reasons, there's these kind of isolated reports like really interest me. And I've been following up on this particular team from the University of Exeter. They've been like collecting, like samples of, people with congenital hyperinsulinism and trying to identify genetic diagnosis, like it happens one by one. And we discussed one paper last year on the hexokinase mutation that causes congenital hyperinsulinism. So now they have an updated report on where they find that CNVs is the cause for this disease in like few patients. And they try to see, okay, patients with very similar regions of CNVs, and then they try to overlap the CNVs to see if they can pinpoint the gene. And it's very interesting because what typically in a workflow, what we do is like we always assume that it is some gene getting deleted, right? Some gene within CNV is actually the one that's causing. And so we will restrict our focus towards genes that fall only within the overlapping regions of the CNV. And that's exactly they did. They triangled out some 5 or 6 genes, and none of them had any kind of link with, like biological link with the hyperinsulinism. And then okay, they decided, okay, let's extend the window a little bit. And then they found this one interesting gene, FOXA2, which has been previously associated with hyperinsulinism, heterozygous loss of function. And it makes sense. Then they went and then they can see that it actually does a couple of CNVs actually deleted partly the coding region of FOXA2. But there are a couple more or 3 more cases where not, right? And it turned out like these CNVs actually deleted the regulatory region of FOXA2, and these deletions have the almost same effect of deleting the whole gene. That is like, this is kind of the thing that we are hoping to look at, find out when we are doing whole genome sequencing analysis in the UK Biobank, right? For now, we have carriers of loss of function mutations in the UK Biobank. One of the things that we hope by looking at the regulatory regions can be increase this number of carriers, right? People with severe deficiency of the gene because of deleting some rare non-coding region who will have similar disease risk effect like deleting the gene or having a loss of function variant. It's going to be extremely challenging, but these are all very nice examples like to go, like see have like an empirical data. It is possible that there are regulatory regions, if it is deleted, that can cause a loss of function like effect and the disease, very similar disease. So that is the FOXA2. And the other one is the ZNF 808. It's slightly different, but again, it boils down to the non-coding region. So this is here, it is a transcription factor, and this is like people from the teams from Exeter, Cambridge, and Finland scientists. And this is like some of the same scientists from the, Exeter also involved in this. And what they found here, so here they are looking at trying to establish genetic diagnosis for, like pancreatic agenesis, people, who are born without pancreas. And so here they identified like 2 patients where they had homozygous loss of function of ZNF 808. And then they did all the functional analysis. It's very interesting because like this gene actually is a transcription factor. And the function of this gene is like it silences all the transposons in the genome. So loss of this gene actually results in aberrant activation of transposons. So our very next topic is tandem repeats. We will come to this, but transposons is comprise 50% of the genome. Most of them are dormant, right? And activation of transposons causing disease is something it's very early stages. There have been like few cases, but that is like a whole area, right? And this kind of falls under that. And the most thing that really interested me here is this gene is actually specific to primates. It's not a conserved it's not a conserved gene. So I don't know if we discussed this during our last, year podcast when we are when you're discussing the whole genome sequencing paper from the decode. One of the interesting finding is, so they did this constraint metrics, like sliding window analysis, where they look at what all the regions in the whole genome where mutations are very less than what we expected. So those are probably disease enriched regions, right? And what they found is almost 50% of the constrained region, the non-coding constrained region, are specifically seen in humans, and it is not absent in other species. So, human specific constraint, right? So this is one, I think the first example I've seen where a gene which is like primate specific actually causing a neurodevelopmental or like a developmental, sorry, not neurodevelopment, developmental disorder causing pancreatic agenesis. So it's a very interesting, like other aspect. The other important, very nice thing they showed is like when they actually, this is important, this gene is important to actually, like control the trajectory of this gene expression programs where decide whether control the fate of these different cells, whether they become liver or whether they become pancreas. So loss of this results actually pushes all the gene expression programs towards liver, and that they also show using functional studies. It's a very interesting paper, but like I'm interested. I was like found this very interesting because of the non-coding aspect of it, and also the constraint, the first primate specific developmental disorder, I think.
Yeah, I think the point about conservation or lack of evolutionary conservation and the novelty and evolutionary time of this gene caught my eye as well, because as I mentioned before, spent a lot of time looking at the non-coding genome, and there are these interesting segments of the genome that are ultra conserved, so they're nearly exactly the same all the way back to fish or flies, or even further in some cases, where, and from the reasons you named, you'd suspect that maybe they are critically important. But then there are other regions of the genome that are called human accelerated regions, which were those that are ultra conserved, but then in relatively recent evolutionary time, they've changed a lot, for example, between humans and primates. And again, there's a question there of, are these human accelerated regions particularly important? But then you mentioned a third here, which is totally novel, at least to humans relative to other primates or other species. And I think one of the things that's been a challenge for the field more generally is understanding the language of regulatory regions. And it'd be interesting to talk about UK biobank and long read sequencing and some of these other points a little later in the show, but we still seem very far from understanding the language of the non-coding region. And I don't know that the whole genome sequencing in the UK biobank will necessarily solve that, because there's probably some kind of cell-specific grammar that we really need to understand through large-scale cell screens and maybe large language models. Thinking of the regulatory space as a language could be an interesting way to think about this as well. But it seems like we're still far from cracking the code of the non-coding genome in the way that we know the triplet code of the coding genome very well. But this is a good example, I think, is the more examples that we find of non-coding regions causing disease, we can start to work backwards from those and understand a little bit. Once we know how it gets broken, maybe we figure out how it's constructed in the first place.
Exactly.
Do you want to transition to tandem repeats? And so the amazing story from around the Huntington's repeat expansion is the first on our list, but I was wondering actually if we save that till last, because I think it's to me, it's one of the most interesting stories of this year. Just from we can finish on that one, and then we'll go into the, not predictions maybe, but themes to watch for 2024. Does that sound good? Let's start with the STR mutation rate paper. That was definitely news to me, having spent a lot of time learning about de novo mutation rate in SNVs in my PhD. I didn't think much about STRs, but we've got the data now to do it.
Yeah. So this is other, really interesting topic area that's going to pick up after, because of this whole genome sequencing data, huge data that we are getting from UK Biobank, right? So this is one area where I'm very excited about. And, if you look at the genome, right, 50% of the genome are repeat regions. So even, if you look at the long document from the Human Genome Project, from the, the publications in Nature and Science, there'll be like very interesting section on how they annotate the genomes. As the first time we have an annotation of the full genome, right? 50% of the genome is like repeat region. Of this, 45% are just transposon. They are like, jumping genes, as we say. And among the remaining, there are like, we have very large, repetition regions. That's what we see in the centromeres and the telomeres. And then there's like pseudogenes, segmental duplications. Then we have this very interesting type of variants called short sequence repeats, right? So it's just the repetition of few one, like two, three, four, few base pairs again and again and again, right? And so traditionally they divide this into two categories. One is like microsatellites and the other one is like mini satellites. So the microsatellites are, I think, like two base pairs up to 10, some people say 7. And these are the ones that actually drove all the linkage analysis that had been happened before we started doing GWAS, because they are very polymorphic, one of the most polymorphic regions. And so you can have this like restriction digestion based markers, which you can use to map the gene disease locus, right? This happened very early, right? And these are the ones that's been also used as a DNA fingerprinting. And so a lot of neurodegeneration related, repeat expansions and things like that, like triplet lipid expansion, they also fall under the microsatellites. Then we have the mini satellites, like more than 10, or this is called as VNTR, variable number tandem repeats. This is something we still don't know. But the interesting thing is, like, there are some that are very common. Like, for example, there's a VNTR in the promoter region of insulin. This is the very first risk gene associated with type 1 diabetes. And then there are a lot of VNTRs that's been classically studied for candidate gene associations. We still have to look at the data to see if those are true, because like a lot of the GWAS disproved all the candidate gene hypothesis that happened before, but the VNTR hypothesis is still there because we haven't looked into that. So that's one thing the UK Biobank is going to help. And so the decode paper is very interesting because, they are typically, all the mutation rate papers come from decode, because they had this very beautiful multi-generation data set where they can actually look into, the mutation rates across the generations. So here they look into the STRs, they particularly look into the microsatellite mutation rate. And I think the most important impressive finding here is, like, we know they are very highly mutable regions. And so they give like an data empirical estimate, right? So it's like they say around 13, like 63 to 65 mutations per person per generation. You need to compare this with the single nucleotide variants, right? So there we are looking at the whole genome, where anywhere they can occur, and it is like 60 to 70, right? This STRs they are looking at is just 1.5% of the genome. And just that percentage alone has that much, produced that many mutations per generation compared to the whole genome SNVs, right? And if you scale it, I think it will, if you imagine a situation where the whole genome is STRs, then every person, every generation, you will have like 2,000 new mutations. That is like amazing. So I don't think anything comes close to it. That's like really, like, it also makes sense. It's one of the most polymorphic site. And the biological reason behind this is also very important, and probably for our discussion about the HTT, right? So the DNA polymerase, when they try to replicate the genome, when they really struggle when they come to this repetition regions, right? So a lot of time this repeat regions form loops, and then when they actually try to fix it, but instead of, like, they get confused and keep going in, they kind of put more than what actually is there, and that kind of results in the expansion. So all this, because of these reasons, that it's very unstable region, and there's like causes hypermutations. And so I think this decode paper will be like a very important reference in that. So that is one. Then we have the other paper from the Laws group, where they actually did some interesting association analysis of VNTR. So this is the the mini satellites that we are talking about. So the very interesting background fact of this paper is that this evolution of data sets from the UK Biobank. First we had the array data, Then we had, sequencing data. Then we are having now like the whole genome sequencing data. And often people, companies fund this, but there was like a competition about who gets hold of the finding, who discovers things first, right? And there are this people like, who are expert in this imputation, like Paul Raw and, people from there, they actually do this like every time just try to do this, before, for example, they did this imputation of whole rare variants even before whole exome sequencing was available. And they found all the like a lot of interesting genes even before we can do it. People did it with the actual data. And it's the very same way they actually recently, last year, they published this repeat regions and the coding regions of the genome. They published two important findings. One is the AKN for the height, and the other one is the LPA. Repeat regions has been known. They gave a lot of new insights, right? And so this year, they have published a paper in the Cell where they look into the repeat regions in the not coding region, focusing on the VNTRs. And the major, take home here, there's like so many interesting analysis here, at least for our discussion here, is that they identified a couple of genome-wide significant locus where the actually causal variant is a VNTR. There's two interesting findings. One is like glaucoma. There's a genome-wide significant locus at near TMCO1. This is one of the strongest locus identified very early in the GWAS timeline. And it turned out the causal variant here is actually a VNTR, 28 base pair variable number tandem repeat. Similarly, colon cancer, I think that's discovered this gene. There is like this genome-wide significant locus at EIF3H gene, and this has been like identified very early in 2008. It seems the causal variant here is like a 27 base pair variable number tandem repeat. So there is like whole bunch of like list of these kind of genes sitting in the GWAS catalog where the causal variants are actually structural variants. There's like a very beautiful paper from Decode last year based on, like long read sequencing where they kind of identified a lot of such examples. We'll be like looking at a lot of these as people start analyzing the whole genome sequencing data. But the interesting thing is there are people more intelligent, more brilliant who can predict this, impute this even without any whole genome sequencing data, just based on, like this haplotype sharing and linkage disequilibrium. And it's really fascinating. And, yeah, it's really very, very interesting paper. I presented it in a journal club in our RGC, actually.
Yeah, I think it is a super cool paper. And for figure 3, if people do want to look up the paper, has just a very clear plot that shows how many repeats someone has, and then the glaucoma risk, intraocular pressure that's around the glaucoma locus. But it's quite a clear effect and a pretty significant one. And these aren't uncommon, 10 a low frequency of 5% or so in some cases, or nearing 5% for some of these repeats. I was going to ask the question around long read sequencing, short read. So I think these papers, they use clever imputation methods, then they validate them with long read sequencing. But I'm wondering, from your perspective, is there a huge amount that's going to be discovered here when we start doing large scale whole genomes using long reads? Or have these been kind of statistically worked out where we shouldn't expect to see, a blossoming of many new micro and mini satellite discoveries that come out of long read sequencing? Or is it the case where we don't know what we don't know until we do some of these studies in a major way?
Yeah, I think it's very difficult to guess that. Definitely, it's, like just using a short read whole genome sequencing, you're not going to find a lot of this, but that will boost some of the, association analysis. But I'm sure like there will be more targeted long read sequencing analysis based on insights you get from the whole genome sequencing data, or some kind of intuition. Maybe this is, like caused by some, structural variations. Speaking of which, now I remember there is this is also a very low hanging fruit locus for frontotemporal dementia. There is one TMM106B locus been found very early on, and we never knew what was the causal variant there. And recently, 2 independent teams actually did this long read sequencing, and then they identified an olive element that is inserted into the promoter of this region, and they showed this is actually the causal variant. I mean, these kind of more targeted, so they only targeted this region, and then they made use of some of the long read sequencing data, and then they beautifully identified the mapped out the causal variant. So these kind of examples we will see more and more, but in a more high throughput way, or I think like it's still far away from now to have that kind of data.
Yeah, makes sense. Moving into the final one of this, which as I mentioned before, is to me one of the most amazing talks I've seen in a while. You tweeted at live at ASHG after a late night after seeing this great talk. I wonder if you could talk a little bit about the Huntington's disease locus, what was presented, and maybe this new model for what's actually happening. And to me, this is another good example of this is a gene that's been studied exhaustively for a really long time, and new technologies are uncovering some pretty novel mechanisms that hopefully point towards therapeutic options in the future. But yeah, I thought it was such an exceptional talk, and it has me thinking more about these repeat expansion disorders, because Huntington's disease is not the only one. We have C9, NORF72, and FTD and ALS, and I think many people around the world are going to be working to see whether this paradigm applies to other diseases. But maybe you can talk a little about the paradigm first before I get over my skis talking about how cool it is.
Yeah, so this is like really interesting. I mean, this talk is from Steve McCroll's lab, and the person who presented is like Bob Hansecker. I think from he like many people liked it. I also liked it really very much. And this is about this Huntington's disease, right? Like you mentioned, we know that the gene the causing Huntington's was discovered long time ago, and then there's been like huge amount of research and understanding, right? So one of the complaints about when we started this podcast, the first part of the episode, I was telling about the time gap between the initial gene discovery and then the first FDA approval, right? So this is like on average 25 years, but it can be really, really long because there are a lot of challenges, right? I think Huntington's is a very good example about, because we know the gene, we know a little bit about how the neurodegeneration is happening. Just for the sake of audience, Huntington's is caused by this triplet repeat like a mutation in the Huntington's gene CAG repeat, and this results in this polyglutamate track in the protein and this neuronal synaptic protein, and this causes toxicity in the brain and results in neurodegeneration, particularly in the striatal region, right? So, and this causes all motor symptoms and dementia and cognitive deficits. Yeah, so here, like what we know about this is like we know there is like this triplet repeat, and and then like the number of repeats each person can have in the population can vary, and people who have typically, I think, more than 37 actually have or like develop Huntington's disease, and we really don't know the mechanism of how like the neurodegeneration happens, and this is like really hindering the drug development. For a lot of time, people have focused on the accumulated repeat protein, like toxic repeat protein, and I think even billions of dollars have been spent on this, and none of the drug actually took off. And only like few, maybe in the last decade, there's been this new insight on the repeat expansion, right? So then people started thinking like you should be focusing on preventing the expansion of the repeats rather than focusing on the accumulated proteins in the brain, right? And this comes from a lot of studies. There's like a GWAS of age of onset of Huntington that actually showed this mutation in DNA mismatch repair gene actually affects the the age of onset of Huntington's. So what here, I think the major important findings that they presented is like they looked into the repeat expansion, right, of this Huntington's protein in the brain in the striatum in the cell type specific manner, right? And what they found is that this repeat expansion has been happening only in the specific subtype of neuron called medium spiny neurons in the striatum, and it is like absent elsewhere. So we know this this is first time they're showing actually in using the single cell technology. And then I think the other major thing here is like they look into how many repeats are there, right? So they found that number of repeats in the brain of these like in this disease patients are like in thousands or a few thousands. And typically our cutoff in a clinically, when we say disease risk for Huntington, is like 37. And what we see in the brains in these patients actually have like far above that. And somehow we know about this before, but this is the first time they're showing in a cell type specific manner. But the important insight here is like how these people who have, let's say, 40 repeats or 45 repeats end up having so many more number of repeats in the brain. So this happens slowly, right? So the age of onset of Huntington is like around 45 years. And so it turns out that, like the somatic expansion happened through two phases. One is like early, very slow phase. And then there is a tipping point, right? Where you actually, the pathology accelerates really fast. And they did some very interesting analysis using the single cell, like data, where they looked at the gene expression profile of different types of repeats, right? So when they actually saw that at particular level, right? I think it's 180 repeats. What happened is completely the gene expression program is disrupted, where you can anymore you cannot even say this is a medium spiny neuron. Even the cellular identity is lost. But before that, before the 180 repeats, there is like not much of an effect, you know. So the idea is that people slowly accumulate, expand the repeats because of this, like the known fact about this DNA polymerase and this how you get this repeat expansion over cell division. And this happens until 180, probably it happens slowly. And then after that, there is like really a fast phase where things get really worse, you know. And so you might have like a window where you actually treat the patient and prevent these things happening. And there's been like a lot of interest in looking at DNA repair genes, MSH2, MSH3 genes as a means to, prevent this repeat expansion. And I think the field is really going to pick up. There's going to be a lot of research into this in the upcoming years. But I think like what Bob Hansick presented, so I don't think I'm presenting all the major findings it did, but it's really a big advancement in our understanding. And this is gonna like really, amazing how this is going to be picking up in the upcoming years.
Yeah, I couldn't agree more. And there's other stories that I'm going to be following that are kind of in the same single cell and somatic, mutation sort of theme. There was an interesting paper that looked at somatic mosaicism in metabolic liver disease. So looking at NASH and, sort of a similar story of understanding at a single cell level how individual changes and then the collection of these changes within the ecosystem of the liver, or the organ, whether it's the liver or the brain or otherwise. And I think it's adding a layer of useful mechanistic information that the germline data just doesn't tell us, right? We know that in this Huntington's case, people start with a particular number of repeats that we can identify based on sequencing in the germline, but what's happening in this particular cell type is something really different, and understanding that is probably the key to treating it, as you've said.
So yeah, there is like one very fascinating article on Huntington's, the whole story in Stat. We should put that in the link. It covers the full story from the gene discovery until now. What are the insights? What are people are really looking into in treating this? So that also covers the findings from Steve McCurl's work, group labs work.
Great. I think I've just found that article. I'll stick it in the notes. So to close out here, I wonder if you could maybe share, you've got 3 or 4 points that I think you're going to be watching closely, 3 things in 2024. The first one is maybe important continuation of the discussion that we've had today, but I'd love if you could just give a highlight of each of these and the kinds of things you're going to be watching.
Yeah. So what I'm going really looking forward into, like 3 major areas. One is the era of genetic medicine. So right, this is the era of genetic medicine. Like many of the findings that we discussed today, we discussed also the translational aspect of it, any drug targets and things like that. And this is how it's going to be given. All the new data is coming up. So I'm really excited about what are the new, rare variants or common rare variants that that give you insight about new drug targets. And yeah, so that's something that I'm very interested in, also because I kind of work in that line, right? So the first one is the era of genetic medicines, obviously. What new discoveries that come up in whole genome sequencing or exome sequencing, etc. And the second one that I'm really interested in, like the technology, the long range sequencing technology, how it's going to improve, our understanding of many of the diseases. So here there's like very, very nice example that I actually highlighted that was a preprint where they showed that they actually solved almost a 25-year-old genetic puzzle, So we have like different types of ataxias, like spinocerebellar ataxia. There is a type called SEN4, SE4, and like this type, there was like first identified in a family in Utah, right? So, and they identified that using the linkage analysis, they identified the genomic locus, but and if you read that paper, the authors are like more optimistic about identifying the causative genes probably in the next few years, but they had no idea that it will be like not identified for the next 25 years. It's a very
Right.
complex region. There is like a lot of segmental duplications. It's a very GC rich enriched region. So it seems almost impossible to resolve the region using short read sequencing. And like these people actually thought, okay, hey, now we have PacBio and all the technologies to do long read sequencing. Why don't we look into these patients? They went back, they looked into the archive, this DNA sample of the very same family, and they did the sequencing using long read sequencing. It's there. It's very clear. They found
Same
there's
family. I didn't catch that it was the same family. That's amazing.
the GGC repeats, in the ZFHX3 region in the exon 10. And it's a GGC repeat, it's a triplet repeat. And it's like they even did some in vitro studies to show if you knock it down, you can actually, reduce all the pathology that caused by this repeat, actually. So it's a very powerful example of what new technology will bring. So this is always the theme for me. I mean, all the things that we do here, for example, people always criticize about we are finding thousands and thousands of genes from the GWAS association studies and things like that. What these genes are going to bring up? I mean, what we are doing is we are producing the data for future generation to work on, So all these discoveries are cumulative. Like we work on all the data that's been generated over the past 10 years or 20 years. Even now, we go back to the genetic linkage studies, family linkage studies, and then we, solve this problem, like this example. So this is how it's so we shouldn't overlook what we are, doing here just because we did not understand. As the technology progresses, we will definitely, people might make big breakthrough discoveries in this. So that's one reason why I'm really interested in the long read sequencing, how it, will change the landscape. And the third one is like phenotyping. I always talk about it. One of the important things we really need to innovate is like how we do phenotyping in genetic association studies. And this is really, really important for conditions like, brain related conditions or psychiatric disorders, neurological diseases. We really don't have like endophenotypes, like, you can just go and do an LDL cholesterol genetic association study. You can find protective genes and you can use this to develop drugs for cardiovascular diseases. It's not that simple for diseases like schizophrenia or bipolar disorder. We need to innovate the phenotyping. One thing that I'm really excited about is going really very proximal to the DNA, looking at, cellular level phenotyping at the cellular level. There's like the very starting of this year, there was a beautiful paper from Soumya Chaudhary from Brigham's, and people from, like a lot of other people from the Harvard. They actually looked into they used the induced pluripotent stem cells to derive, like cells, and then they did some phenotyping of, cellular, how the cytoplasm or the mitochondrial distribution. They used this microscopy. And then they did a genome-wide association study and exome-wide association study. They found beautiful association signals. They found three peel-off signals that is like a actin cytoskeleton genes that actually, when you have loss of function of that gene, it disrupts the shape of the cytoplasm. And there's like a prolactin receptor gene that actually disrupts how the mitochondria is distributed within the cells. And this is a fantastic proof of concept. Like this handful of iPSC cells, like handful of samples, and you can find these associations. So by going into the cellular phenotyping, you can really increase the power And this is what is going to change the landscape of, the field like psychiatric genetics and things like that. And already people are working on those things, right? Trying to use the psychiatric patients, cells like to do this induced pluripotent sensors, derived neurons or differentiate them into neurons or things like that, and look at all the cellular level, phenotypes and associate them with their genetic factors. So that's like really I'm excited about. And you can also, one other area where I am kind of curious is like how people will use the embryo samples, the embryo screening is picking up the recent ORCID announcement and things like that. And one untapped potential there is like, all these IVF clinics, they discard a lot of embryos, and these embryos can be an amazing sample to do establish some genetic, variation database and things like that. There's like very nice pilot study, a preprint where they actually looked into the discarded embryos. Often they get discard embryos for reasons like, if the embryos have aneuploidy, like trisomy or monosomy and things like that. So they collected all such embryos and then they compared with the normal embryos and then did this genome-wide association kind of study. And they identified a very interesting locus. There's like a microtubule gene, which is actually the main gene important for the chromosome segregation during this, this meiosis, right? And that is the major reason for this, like those anomalies causes this aneuploidy. It's a fantastic proof of concept where you can use this kind of samples to do that. I mean, from a population genetic standpoint, I think like all our databases right now, we have was like adult samples, elderly samples, like middle-aged samples, and we really lack pediatric sample, but we also lack, samples. We should go even beyond before birth, like prenatal, any kind of samples from the prenatal. This will really give you, idea about, all these constrained genes that causes, prenatal mortality or postnatal mortality. There's a beautiful paper from the Decode where they look into the homozygous depletion, the peel off, the peel off, the statistics, PLI statistics that we use to define genes constraint, from the nomad is based on the heterozygous loss of function variance, right? We don't have anything for the homozygous loss of function variance. And that is one area people are really interested in that. And to look into those things, you really need databases that go, to pediatric samples or even, like newborn or even embryo samples. I think there's like a lot of untapped potential, both from the academic side and also from the, like industry side. And I'm excited about that. I know some people are interested in that area. They are going to do some great, very fascinating stuff there.
Yeah, absolutely. Those are all great areas to look at. As always, second year in a row, love this episode. I think we've covered an enormous amount of ground. We discussed that we'd probably hit somewhere close to the 3-hour mark on that, but that's okay. But it gives people something to do over the holidays, catch up on anything that they've missed in 2023. I am always amazed by your encyclopedic knowledge of everything that goes on in this field, and also the great storytelling that you do. I think at the heart of all of these is a really interesting cast of characters and people who focused on some corner or giant open plane of new biology that they could uncover. And I think you do an amazing job telling the story about not just the new findings, but the people behind it that are able to figure these things out. So thank you as always. And I'll just give a very quick plug again that I think everyone, if they're on Twitter, should follow you. He's at Dr. Veera. And also you've got a Substack, GWAS Stories, right? They can go to gwasstories.com and subscribe if they want to get the written version of this and anything else. Anything else that you're looking for if you want to do a quick shout out for anything?
No, I just want to just thank you again for this opportunity. And like I mentioned before, I'm a big fan of your podcast series, and I think it like a lot of people are really missing on that. There's like huge amount of information there, things you can hear from people only. It's available there. I think it's amazing what you're doing here. I still see how couldn't understand how you are finding the time when you're also running a company, as a CEO. But it's really impressive. And yeah, thank you so much for this opportunity. Hope I didn't take like too much time.
No, it's great, and it's my pleasure. It's some of the best learning I can do, and we're actually, maybe this is a good time to announce we're moving to weekly in 2024. So up till now we've done bi-weekly, but we've had an amazing set of guests in 2023. We've got many more exciting ones in 2024 coming. So yeah, it's
Exciting.
fun. It's one of the best hours or a couple hours a week that I get to spend. So thank you, and happy holidays. Great to see you.
Thank you very much. Happy holidays to you too.
Great. And thanks everyone, as always, for listening. We hope you enjoyed this special 2-part episode. Please give us feedback if there are any things you wish you would have heard about or guests that you'd like to see. And to all of you, thanks so much. If this is your first episode or if you've been listening since the very beginning, hope you have a happy holidays, and we will see you in 2024.